Is your pH meter lying to you? Before you trust that pH reading, here's a scary question. What if your pH meter has been lying to you this whole time? Dramatic pause. I've seen students fail experiments and researchers waste months of work because of uncalibrated pH meters. Today, we'll explore professional pH meter calibration steps, prepared by chemical engineer Weynseht Fereadea. Did you know an uncalibrated pH meter can be as unreliable as a broken thermometer? Before you trust your next measurement, let's ensure your device is perfectly calibrated. Why calibrate? Think of your pH meter like a bathroom scale. If it's not zeroed properly, every measurement will be wrong. Uncalibrated meters cause inaccurate data that can ruin research, wasted chemicals and money, failed quality control checks. Calibration resets your meter using solutions with known pH values. No calibration? You might as well be guessing. What you need? You'll need 1. Your pH meter. 2. Fresh buffer solutions. I'm using pH 4 and 7 today. 3. Distilled water. 4. Clean beakers or measuring cylinder. Pro tip: always check your buffer expiration dates. Proper pH meter calibration procedure. 1. Preparation. Ensure your pH meter is clean and properly maintained. Gather fresh buffer solutions, pH 7.01, and 4.01 or 10.01 if available. Prepare clean beakers or measuring cylinder for each buffer solution. 2. Rinse the electrode. Rinse the pH probe thoroughly with distilled water. Gently blot dry with lint-free tissue, never wipe. 3. Calibration with pH 7.01 buffer. Pour 20 to 30 milliliters of pH 7.01 buffer into a clean beaker or measuring cylinder. Immerse the electrode completely in the buffer solution. Press the Cal or calibration button on your meter. Wait for the reading to stabilize, typically 30 to 60 seconds. The display should show exactly 7.01, plus or minus 0.02, when calibrated. Press Enter or Confirm to accept the calibration point. 4. Rinse and prepare for pH 4.01. Remove the electrode and rinse again with distilled water. Blot dry gently with clean tissue. 5. Calibration with pH 4.01 buffer. Pour fresh 20 to 30 milliliters of pH 4.01 buffer into a clean beaker or measuring cylinder. Immerse the electrode completely. Press Cal button again. Wait for stabilization, 30 to 60 seconds. The display should show exactly 4.01, plus or minus 0.02. Press Enter to confirm. 6. Optional third point, pH 10.01. If using pH 10.01 buffer, repeat the process. Rinse electrode. Immerse in fresh pH 10.01 buffer. Calibrate and confirm. 7. Final steps. Rinse the electrode thoroughly with distilled water. Check the calibration slope, should be 95 to 105%. Store the electrode in proper storage solution. Important notes. Always use fresh buffer solutions. Replace every 1 to 2 months. The exact button names may vary by pH meter model. Temperature compensation is recommended. Never let the electrode dry out between measurements. Always start with pH 7.01 before moving to other buffers. This corrected procedure ensures accurate calibration following standard laboratory practices. The sequence, 7-4-10, is important for proper electrode conditioning. Pro tips. 3 mistakes that ruin calibration. 1. Wiping the probe, always blot gently. 2. Using old buffers, they expire. 3. Forgetting temperature, buffers and samples should be the same temp. Protect your electrode. Store in KCl solution. Heavy multiplication X never exposed to dry air. Clean monthly with 0.1 m HCl. Heavy multiplication X avoid touching the glass bulb. Now you'll never question your pH readings again. Smash that like button if this helped, subscribe for more lab hacks, and comment below, what experiment will you use this calibrated meter for? Thank you for joining us on this tour of pH meter calibration. Always remember, precision starts with proper calibration. We'll see you in the next video.